Well, hello again, everybody. We are back in our looking at the Bible and the way that it touches on areas of creationism and philosophy. And today we're going to be looking at DNA and how it replicates. And this is just part of where we started with the last segment where we looked at abiogenesis. So it's sort of carrying on from that because I wanted to show the intricacies of DNA and what it does, because it's just amazing. It really is amazing. Every time I teach this in class, I want to point out the evidence of design, and so I think this is an important thing for us to look at in this series. So first thing we want to do is describe the DNA molecule. Here's a nice 3D image that you can get from Wikipedia for free. You can see that the DNA molecule is a double helix. Think of a ladder that has two sides, and then it has the rungs on the ladder, and then take the ladder and sort of twist it. And that's what the DNA molecule would look like. DNA was actually discovered, or not discovered, it was first, a picture of what it actually looked like. Its structure was first discovered in 1953 by James Watson and Francis Crick. And they actually used a technique that was developed by Rosalind Franklin called X-ray crystallography. Now, just a little interesting side note here, a historical side note. Turns out that when Watson and Crick, they discovered this molecule on a Saturday, and when they went to the pub afterwards with everyone from the lab to celebrate this amazing discovery. One of the most famous statements that were made were that they said that we have just discovered the secret of life. Now, I thought it was pretty cool. And it would be an amazing discovery to see this incredible complex molecule that is responsible for us, for who we are. So here is the DNA molecule. It's unwound in this picture, of course. And the DNA molecule is made up of nucleotides, something called nucleotides. Here are 6 nucleotides that are circled, and we'll talk about what they are here briefly. So here's a nucleotide. A nucleotide has a phosphate group, so obviously it has phosphorus in it. It has a sugar group, a pentose sugar, which has carbon in it. And it has a nitrogenous base, because the nitrogenous base has nitrogen. That's the fundamental part of that particular base as well. So they're the 3 chemical molecules that are used in a nucleotide. And a nucleotide is a monomer, and by that I mean monomers get together to form some sort of complex molecule. So proteins, the monomer for proteins is amino acids. So amino acids get together, you get a protein, and in order for a DNA to get together, you need these monomers of nucleotides. Okay, so with DNA, there are 4 bases. 4 nitrogenous bases. They are adenine, guanine, thymine, and cytosine, given the letters T for thymine, A for adenine, G for guanine, and C for cytosine. And you can see in this picture they're given different colors, and you can see the letters on them as well. With these 4 bases, they pair in a complementary way, which means that adenine always pairs with thymine and guanine always pairs with cytosine. So again, looking at the picture, you can see the thymine nitrogenous base is pairing with adenine, the guanine is pairing with the cytosine. The 2 strands are anti-parallel, and this is really, really important. What does that mean? Well, if you look at the strands, if you look at the nucleotides over here, you can see that that sugar, okay, its point is going upwards, but on this side you can see the point is actually pointing downwards. In other words, you got the same sort of structure on this side, but it's reversed on this side. And I'll get into why that happens shortly. But it's really important because they give each end of these strands a name. They call this end the 5' end and this end the 3' end, and it's named after the carbon on that sugar. So notice here's a sugar, the pentose sugar. You've got a carbon on each one of these edges here, on these corners here I should say, but you've also got a carbon pentose sugar. And that 5' carbon the 5' carbon is pointed in the same direction that the point is on this sugar, and so they've given this direction that name. They call it the 5' end because of this carbon, and in the same way they've given this end the name because of this carbon, the 3' carbon. I mean, I guess they could have called the 2' end as well, but they called it the 3'. So you have a 5' end, which is this end of the molecule, and a 3' end, which is this end of the molecule. So you've got nucleotides going in that direction, and you can see that by the pointy part of the sugar, and you've got nucleotides that are going in this direction as well. And we'll talk about that momentarily. We want to talk now about semi-conservative replication. What does that mean? Well, if you've got a DNA molecule, so here's a DNA molecule, it's wound up, it gets unwound, we'll talk about that in a minute, and then you're left with two halves of your DNA molecule, your double helix, you're left with two halves. Well, what happens is they are used as templates for a new strand of DNA. And so that DNA is attached, that strand I should say, is attached to this strand, and so you end up with a whole new double helix on this side and a whole new double helix on this side. So there's your two new strands. But notice that both strands have one half of the strand that comes from the original molecule. So that's what it means by semi-conservative. All right, DNA replication. So this is all the fun parts. So what happens? How does DNA replicate? And there are lots and lots of enzymes, or I like to call them nanomachines, because it's really what they are. They're very complex little molecules that are used in replication, but the three biggest ones, or the most important ones, are helicase, polymerase, and ligase. And so let's have a look at what each one of these does. First of all is the helicase, and so that's represented here by this blue oblong shape. So that's the helicase. The helicase attaches, it actually unwinds the molecule, so it physically unwinds the molecule, and then it also chemically cuts right in between these bases, right along here. So between the two bases, it'll cut a chemical cut, and that will cause the DNA molecule to unwind into two separate strands. Now that we have two separate strands, now we can add new nucleotides to it, but this is where it gets a little bit complicated. By the way, this is called the replicating fork right here, because this is where all the action happens. So remember we talked about the 5' and 3' ends. Well, it turns out that this polymerase, DNA polymerase, it can only attach to the 3' ends of these strands. In other words, it has the ability to attach to the 3' end of the sugar, but it doesn't have the ability to attach to the 5' end of the sugar. It's been made to attach just to this side. And that's fine for the leading strand. So this is the leading strand, and this is the 3' end of the leading strand over here. Well, that's great, because it means the polymerase can just attach itself to the 3' end, and then it just literally just zips along the strand, adding these nucleotides until it meets and gets up to the replication fork. That sounds great. But what's the polymerase going to do when it wants to add nucleotides to this strand called the lagging strand? Remember, it can only attach to the 3' end. And in this strand down here, this end is the 5' end. If this is the 3' end, this is the 5' end, which means the 3' end's down here. And so literally what it has to do is it actually has to attach down in the replicating fork, and it works in the other direction. So I mean, it's actually working in the same direction literally, because it's going from a 3' to a 5' end. So it's going in the same direction, but it has to go away from the replicating fork. And while it's going away from the replicating fork, what it does is it builds nucleotides in that direction. So it will build nucleotides in the direction away from the replicating fork, but then once it reaches a fragment that has already been created, it has to detach from the strand, has to go back to the replicating fork, reattach, and then it will build a new fragment up to where it finished the last time. So hopefully all of that makes sense. And that's because it has to attach to that 3' end. Well, that means that when it's making these nucleotides, it has to make them in short segments because it has to detach and reattach behind where it started the last time. And these fragments are given a name. They're called Okazaki fragments. The third most important enzyme is the ligase. So the ligase then will zip along both strands and will connect any broken pieces along the way so that the strands are perfect. Okay, so that is how replication occurs. So let's have a look now at all of this in action. So I'll just play it through, see if you can see what's going on, and then we'll sort of slow it down and have a look at it in a little bit closer detail. This is really cool. Okay, so now let's slow it down here in this next video and blow it up. So here's our DNA helicase, and I'll play this through a couple of times. It's splitting the DNA molecule into two. There's one strand going this way, there's another strand going that way. This is the replicating fork. Here is a DNA polymerase, and this one is on, well, see if you can figure it out. What strand is it on? Notice that the DNA polymerase, even though it's the actual strand going through the polymerase, the DNA polymerase isn't moving. You can think of the polymerase as working towards the replicating fork. So what strand is this? If you said the leading strand, you would be correct. So that's all this polymerase does. It sits on that molecule working towards the replicating fork. Let me go back again to the beginning. It sits on that molecule working towards the replicating fork, just adding nucleotides, which means that this side, well, this is the lagging strand. So what happens here is you can see the DNA polymerase, but it's building nucleotides in this direction. Here's an Okazaki fragment that it had built previously. So it works up to that Okazaki fragment, and then it detaches and reattaches down at the beginning of the replicating fork, and then it works back up to the Okazaki fragment that it just finished. And so that is how you end up with two strands of DNA. So I want us to look at this now in all humility and ask ourselves what we see. I mean, what do we see? Do we see just a clunky sort of completely random bunch of molecules that are just randomly doing something, or do we see evidence of design? I mean, I think if we are honest, we've got to say to ourselves, I see design in this. I see that all of these parts play a role. They have a function, and they're performing a task. And not only that, all of these tasks are interrelated. One task can't occur without the other task. This is absolutely the foundation of information. This is very complex. Now, as a creationist, I know that there are issues, many issues in creationism, and lots of them that I don't have answers to. But one thing I know for sure, this is a no-brainer when it comes to intelligent design. Now, I know that some opponents of intelligent design will actually look at this and tell you that this is actually evidence of poor design. And the reason they do that is they say, well, we think that the polymerase should work towards the replicating fork on both strands. We think that the lagging strand and the addition of these Okazaki fragments is actually evidence of poor design. It's clunky. It's really not optimal. But is that really true? So here's a paper that was published recently in Science Advances, and in that paper the authors actually found a tRNA molecule where the RNA polymerase actually adds nucleotides in the well, it jumps onto the 5' sugar and works towards the 3' end. And that's opposite the way it does in DNA. So this is really rare for this to happen. But what they found lots of interesting pieces of information as they were looking into this particular process. First of all, they found out that it's actually a lot more complicated than going from the 3' to the 5' end. In other words, for the polymerase to jump onto the 3' sugar or end and work towards the 5' end, that's a lot simpler than it is when the polymerase has to work the other way. It requires an additional enzyme with 2 extra active sites that have to be lined up precisely. It also requires a lot more energy, is what they found out. So that's really interesting. It turns out that therefore it's actually more complicated to work in the direction where the polymerase will jump onto the 5' end and work towards the 3' end. It's a lot more simpler for it to work from the 3' end to the 5' end. It requires less enzymes, it requires less energy. Now, translate that over to the DNA molecule and remember that there are 2 strands being made. That means if you were going to have the polymerase working in 2 different directions, because that's what it would have to do in order to both work towards the replicating fork, you would need then 2 times of everything. You would need double the amount of enzymes and proteins at work. That means you've got to fit all of that nano machinery into that very tiny space in the replicating fork. Well, that doesn't make any sense. That would be very cumbersome and complicated. So it makes more sense for the DNA polymerase to work in a single direction and then simply to attach and deattach like it does on the lagging strand. It would also require a lot more energy to do that. So there are good reasons why the polymerase works away from the replicating fork on the lagging strand. And so this idea that it's poor design, that's simply naysayer talk. Don't believe it. And typically this happens in areas that are opposed to intelligent design. Typically people are very vitriolic against it. And so they want to find so-called evidences of poor design. Yet let's go back and look at this molecule in all humility, and can you honestly say this is evidence of poor design? I think this is where scriptures like Romans 1 come into play, where the unrighteous, they suppress the truth because they are committed to a particular paradigm. As I said earlier, look, there are problems in creationism. There are. But this is a no-brainer. And when we suppress what is quite obvious, I mean, in Romans 1, it talks about the fact that it actually receives wrath, that the wrath of God is poured out on man because of that. Of course, at the end of the day, and you'll hear me say this from time to time, ultimately, it doesn't depend on scientific evidence anyway. Ultimately, I mean, Jesus says if someone rises from the dead, they will not believe. Then why would they believe if they saw this? Okay, the fact is that you can only believe that this is intelligent design if you believe in the Creator. And that is a presuppositional commitment that one needs to make, where the Holy Spirit is at work to convince. There's that passage from Romans 1. For the wrath of God is revealed from heaven against all ungodliness and unrighteousness of men, who by their unrighteousness suppress the truth. For what can be known about God is plain to them, because God has shown it to them. For his invisible attributes, namely his eternal power, divine nature, have been clearly perceived ever since the creation of the world in the things that have been made. And so just going back once again to the last segment we did on abiogenesis and the nature of design. In that example, we talked about 30 people from your high school days, let's say 20 years ago, all of them turning up at the same time in a restaurant in a little town in the middle of the US at the same time. Think about that. 20 years displaced from these people, and everyone just turns up in the same restaurant in some, backwards little country town in America somewhere. If that occurred, you wouldn't say to yourself, oh, what an amazing coincidence. You wouldn't attribute that to coincidence. You'd attribute that to design. You would know that someone prepared a surprise high school reunion. You'd know that something like that had to have happened. It's intuitive. But I also want to suggest that it's actually scientific to come to that conclusion, because nature doesn't randomly produce complex and specified information. It doesn't. Everything we know about nature is that it does not produce complex and specified information. Scientists have been prodding away at molecules for 60 years, spending billions of dollars, and we know that. It's a scientific reality. That doesn't happen. So that means then that I can come up with a hypothesis. And the hypothesis would be is that if natural objects or events, so for example, the 30 people getting together in that little hotel in the middle of America, if natural objects or events contain or require complex and specified information, then, okay, it's a hypothesis, I know that this was specially designed. That's a hypothesis. It's testable based upon what we know about science. And that is exactly the case with the 30 people getting together in that room. It's not just intuitive. You can apply science to that. So join me next time. Next time we're going to look at the actual information in the cell. We're going to look at the actual code involved in replication and in protein synthesis. Okay, so that is it for today. Again, please be praying for me. As always, I appreciate prayer. I think that in reality, Jesus says, without me, you can do nothing. And as I've said before, it's not about the science, guys. It's about Christ. This is a tool. All of this is a tool. And so pray for me, pray for this ministry, pray that people's hearts would be humbled and that Christ would be exalted. Of course, submit to the channel, press the like button, ring the bell, all those fun things to do to get the YouTube algorithm working on my side. Leave comments as well if you enjoyed the video or you have questions, and we'll see you next time. Thanks. Bye.